So for this slide, I will explain about introduction of CRISPR. First, a gene editing technology that allows scientists to cut, remove, or correct specific gene in DNA with great precision. In the context of cell therapy, CRISPR is used to modify a patient's cell so that those cells can treat or prevent disease. In cell therapy, cells, for example, stem cell or immune cell, are usually taken from the patients. Then the gene inside those cells are editing using CRISPR in the lab, and after that, the modified cell are reintroduced into the patient body. The purpose is to repair damaged genes, change cell functions, or make the cell more effective at fighting disease. Next, I will explain about CRISPR Cas9 gene editing mechanism. As you can see in this slide, CRISPR Cas9 functions as a precise gene editing system where guide RNA direct to the Cas9 enzyme to a specific DNA sequence. Cas9 enzyme cut the DNA at the target location. The cell repair the cut DNA through NHEJ, gene count or the activations, HDR, corrections of replacement of fault gene. Cell material interactions. After gene editing, cells are combined with biomaterial scaffold in tissue engineering. CRISPR modified cell can express higher level of cell adhesion molecule. This improves cell attachment, spreading, and proliferation of the scaffold surface. In cell material interactions promote organized tissue formations. Biodegradation of biomaterial. Biomaterial used in cell therapy are designed to be biodegradable. CRISPR modified cell can regulate enzyme productions that influence scaffold degradations. The degradations rate match the rate of new tissue formations. As the scaffold degrades, it is gradually replaced by natural extracellular matrix, or we call it ECM. Mechanical properties. CRISPR edit cell respond more effective to mechanical cues through mechanotron seductions. Different stiffness level can guide stem cell differentiations. Stiffness material, bone cell. Softness material, neural cell. Biochemical properties. CRISPR can enhance cell sensitivity to biochemical signal, growth factors, chemical composition of the scaffold. Edit cell may produce higher level of ECM protein. In this slide, I will explain about application of CRISPR, which is gene therapy, sickle cell anemia, or beta thalassemia. This disease occurs due to mutations in the hemoglobin gene, causing red blood cell to multifunctions. Type of gene therapy use. Gene addition therapy. A functional beta-globin gene is added into the patient's stem cell using viral vector to deliver the genes. Modified cell produce healthy hemoglobin.
Today, I will discuss CRISPR gene editing for cell therapies, focusing on how this technology supports environmental, economic, and social sustainability in modern healthcare. Traditionally, many medicines are produced through large-scale chemical synthesis, which require high energy input, extensive use of organic solvent, and complex manufacturing infrastructure. As drug demand increases, this approach generates significant chemical waste and carbon emission. In contrast, CRISPR-based cell therapies rely on living cell as therapeutic factory. A single carefully edited cell can be expanded outside the body and reintroduced into the patient, potentially providing a long-term or even permanent cure. This reduces the need for continuous drug production and repeated treatment. A key sustainability advantage of CRISPR lies in its programmability to target a new gene. Only the guided RNA needed to be changed rather than redesigning the entire protein system. Compared to other gene editing tools such as TALEN, this significantly reduces material use, cost, and laboratory waste. Moreover, CRISPR-based cell therapies are also more energy efficient. Most clinical applications use ex vivo gene editing, where cells are edited in optimized bioreactor under a controlled condition. These systems are far more efficient in terms of energy and resource use compared to in vivo approach, which often require large quantity of viral vector or nanoparticle for delivery. In addition, the use of closed and automated manufacturing platforms such as those for CAR T-cell therapy integrates cell selection, editing, and expansion into a single system. This increases yield, minimizes contamination risk, and reduces reliance on energy-intensive cleanroom and sterilization process. Another important sustainability aspect is waste minimization for genetic disease such as sickle cell anemia and beta thalassemia. CRISPR aims to deliver a single curative treatment and avoiding the long-term waste generated by chronic care, including repeated drug packaging, consumable, and hospital visit. Furthermore, the high precision of CRISPR, especially with advanced like base editing and prime editing, reduces off-target effect and failed treatment. Fewer failed procedures mean less reagent use and lower overall medical waste. CRISPR also support recyclability and cleaner manufacturing design. The use of less toxic reagent, biodegradable or recyclable plastic, and modular device with reusable hardware and lightweight disposable component helps reduce regulated medical waste while maintaining sterility and safety standards. Finally, CRISPR contribute to long-term social and economic sustainability by preventing disease rather than managing symptoms. By correcting disease-causing mutation earlier, CRISPR can reduce severe complication, long-term hospital workload, and healthcare costs. For example, CRISPR-Cas gene editing in sickle cell disease has been shown to effectively correct harmful mutation, potentially preventing complications such as organ damage, stroke, and recurrent pain crisis. In conclusion, CRISPR-based cell therapies represent a shift towards sustainable biology-driven medicine, reducing energy use, minimizing waste, improving efficiency, and deliver long-lasting benefit to both patient and healthcare system.
There are many ethical and social value that we can get from CRISPR gene editing for cell therapies, and some of them are first, biocompatibility and patient safety. CRISPR enables precise genetic modifications, reducing off-target effects and also improving the safety of engineered cell and tissue for clinical application. Second, disease prevention and therapeutic benefits. The technology allows correction of genetic mutation at the cellular level, offering a long-term treatment or potential cures for inherited and also chronic diseases. Third, animal welfare, which is the reduction of animal testing. CRISPR edited human cell and tissue models reduce reliance on animal experiments, supporting the ethical research practice which is aligned with the 3R principle. The next one, the equitable access to healthcare. CRISPR has the potential to lower treatment costs through scalable and efficient gene editing processes, which is improving the access to advanced therapies if fairly regulated. And last but not least, ethical governance and social responsibility. With proper regulations such as ethical review and also transparent clinical trials, it is essential to prevent misuse and also ensure informed consent and protect the human right.
So next I will be explaining about the CRISPR technologies that align with the UN Sustainable Development Goals, which goals 3, 9, and 12. So first of all, we will go through goal 3, which is to have a good health and well-being. So apart from cancer, there are a few more genetic diseases such as Alzheimer's, Huntington, diabetes, sickle cell anemia, which are caused by mutation in relevant genes. Recently, CRISPR was found to be successfully employed in editing genetic switches, and many microRNA or miRNA are involved in cancer progression and development that can be specifically targeted with CRISPR genome editing system. Target 3.4 of SDG3 is to solve diseases like cancer, cardiovascular, diabetes, and chronic respiratory diseases. So CRISPR is being used to supercharge cancer immunotherapies. For example, it is being used to edit a patient's own T cells, which is a CAR T therapy, to make them more effective in recognizing and destroying cancer cells. So it has been proven CRISPR is able to heal or to help patients in fighting cancer, in fighting respiratory diseases, cardiovascular, and that makes up the alignment with the UN SDG 3. So next, we'll go to how CRISPR technology can contribute to industry, innovation, and infrastructure. First of all, we'll go to agriculture industry. So the first report of CRISPR use in agriculture industry arise from 2013, expected high potential due to the possibility to introduce targeted modifications without integrating random genes or causing random mutations. Recently, India has approved its first genome-edited, high-yielding and climate-resilient rice varieties which were created using the CRISPR technologies. So at present, 9 crops were developed using new breeding technique, NBT, and a few through CRISPR. So CRISPR is one of the technique under the new breeding technique, and from the 9 crops, a few was from CRISPR, which is a GABA tomato and waxy corn in Japan, mustard greens in United States, and non-browning bananas in Philippines. So How CRISPR made this possible is they targeted the only needed genes in the RNA to ensure that there are no mutations happen while they are improving the quality of the crops. For example, for example, they are producing non-browning bananas, mustard greens in United States. So it is contributing to the SDG 9. Next is the innovation. So how can CRISPR contribute to the innovation of the world? So CRISPR is an innovation itself that has accelerated scientific research and development across various fields, directly contributing to the spirit of SDG 9 objective in fostering innovation. So as I said, CRISPR itself is an innovation, therefore contributing to the SDG 9. And the last one is the CRISPR technology in aligning with the SDG 12, which is in sustainable consumption and production. So how can CRISPR have the potential in achieving SDG 12? So first of all, target 12.3 is to reduce food loss and waste. So crops can be engineered to be disease resistant and increases resilience rate from drought or pests. For example, tomatoes or apples may be engineered to resist browning or spoilage. So that's the first one, is to reduce the food loss. So by doing this, whenever a crop is produced and they are engineered to resist browning, they will can take up maybe to 1 month before it gets bad. So it's good innovation and sustainable consumption. So secondly is the in sustainable biofuel and biomaterial production. So CRISPR optimizes microbes and algae for industrial applications, turning waste into renewable products. For example, Fusarium venenatum or engineered fungi become efficient microprotein sources with up to 61% lower environmental impact. And to conclude our presentation, so genome editing has been a transformative effort in context of sciences and human medicines, offering unprecedented opportunities to methodically cut up complex biological processes and treat many chronic diseases such as Alzheimer. So CRISPR is being an indispensable tool in biological research. So CRISPR once known as the bacterial immune system against invading viruses, the programmable capacity of CS9 enzyme is now revolutionizing in diverse fields of medical research, agriculture, and biotechnology. The ability to change DNA bases at precisely determined locations hold a great value not only for any molecular biology but also for medicine and biotechnology. So therefore, introducing desired changes into genomes has been a long sought after goal in molecular biology. Finally, CRISPR holds one of the biggest potential in implementing innovative technologies into medical field, not just avoiding the symptom, but rather curing the diseases itself.